All right, today we're doing something really cool. We're stepping inside the virtual drug lab to see how scientists are using some seriously powerful computers to design medicines that work directly with our DNA. This isn't science fiction, it's happening right now. Let's get into it. So right off the bat, you're facing a huge question. How in the world do you design a tiny molecule that can navigate the incredibly complex, twisted ladder of our DNA, find the exact right spot, and do its job without messing everything else up? It is, without a doubt, one of the toughest puzzles in all of modern medicine. And you really have to get it right, because the stakes are incredibly high. You see, when a molecule binds to your DNA, it can be a good thing, a lifesaver, like the cancer drug 5-fluorouracil that just stops tumors in their tracks. But it can also be a really bad thing, like a pollutant that damages your genetic code and causes cancer. So yeah, this is literally a matter of life and death. So here's our game plan for today. We're going to start with DNA itself, the target. Then we'll pop into the virtual drug lab. After that, we'll check out the amazing computational tools they use, look at the ultimate challenge of predicting a perfect match, and finally, see how all this computer code actually turns into real-world cures. Okay, first up, DNA as a drug's target. You know, it's like they say, if you want to design the perfect key, you have to understand the lock. And for these drug designers, the lock is our DNA double helix, and it has some very specific places where a key can fit. One of the main ways drugs do this is through something called intercalation. And what's wild about this is just how physical it is. You have these flat molecules that literally just slide in between the rungs of the DNA ladder. Imagine sliding a coin into a stack of other coins. It forces the whole structure to stretch and unwind, which totally disrupts its ability to function. The other way is called groove binding. Now, this is a bit more subtle. Instead of forcing its way inside, the drug molecule just sort of nestles into one of the grooves on the outside of the DNA helix. It's a much gentler approach, and it doesn't bend the DNA out of shape nearly as much. But here's the thing, not all grooves are the same. The major groove is bigger, but it's often already occupied by our own body's proteins. The minor groove, on the other hand, is smaller, which allows for this really snug fit. It creates better molecular stickiness, what scientists call van der Waals forces, making it the perfect parking spot for a lot of drug molecules. So we know how drugs can bind, but finding the right one out of millions of possibilities, that's a whole other problem. And that's where the virtual drug lab comes in. This is where we shift from just mixing chemicals and hoping for the best to actually designing them with purpose. And the power of doing these studies in silico, which is just a fancy way of saying on a computer, is just massive. Think about it. Making and testing just one potential drug in a real lab can take forever and cost a fortune. But in a simulation, scientists can test thousands of virtual drugs in no time, cutting way down on the guesswork and seeing things at a molecular level that you could never, ever see in a regular lab. Okay, so how do they pull this off? Well, they have this amazing set of computational tools. You can think of it like a scientist's virtual toolkit, where each instrument is designed to do a very specific and very powerful job. The first tool in the box, and the most fundamental one, is quantum mechanics. This is basically the ultimate magnifying glass. It lets scientists zoom all the way down to see the electrons that form the chemical bonds and drive all the reactions. It's as deep as you can possibly go. And this is where it gets really practical. With quantum mechanics, we can understand exactly how a pollutant becomes dangerous inside our bodies. And we can model precisely how the platinum atom in a famous chemo drug like cisplatin actually bonds to DNA, basically gumming up the works for cancer cells and stopping them from dividing. All right, next up we have molecular dynamics. Now, if quantum mechanics is a magnifying glass for a single moment in time, think of this as a super high-speed, slow-motion camera. It creates a molecular movie, showing you how all the atoms wiggle, jiggle, and interact with each other over time. And with these movies, scientists can calculate the single most important number in drug design, the binding energy, that tells you how tightly a drug will stick. For instance, these simulations showed why two almost identical drugs have very different effects. It all came down to tiny differences in their flexibility and how they interacted with water molecules around them, details you'd only ever catch with a molecular movie. Our third tool is probably the most well-known one, molecular docking. You can think of this as a master key maker. Its whole job is to take a huge library of virtual molecules and, one by one, try to fit them into the DNA's binding site. It's like trying thousands of keys in a lock, super fast, to see which one fits best.
And I mean, just look at this. This isn't just one piece of software. It's a whole mature field of study. You've got programs like Doc, AutoDoc, Gold, Glide, all of them working to solve the same puzzle. It just goes to show you how vital this technology has become.
And that brings us to the ultimate challenge, the holy grail of this whole field, trying to predict that perfect match completely from scratch. This is called ab initio docking. It literally means from the beginning. And the goal is just incredible. Can you just give the computer a drug and a strand of DNA and have it figure out if, where, and how they will bind with absolutely no hints? It's the final frontier. So how's it done? Well, one way is with something called a Monte Carlo simulation. Here's how it works. First, you just drop the drug and the DNA into the virtual world. Then, the computer just starts trying things out. The drug explores millions of random positions all around the DNA. At every single step, it tests the energy. Is it better in the groove? What about intercalating? And after trying out this mind-boggling number of possibilities, it finally locks on to the one spot that is the most stable, the best possible fit. It figures it all out on its own.
Okay, so we've been deep in the world of code and computation, but what does this all actually mean for us, for medicine? Let's zoom out and look at the real-world impact.
This quote from Dr. Vidya Kothakar really says it all. All this work on computers isn't just for academic papers. It saves an incredible amount of money and time in the real world, making the whole process of discovering new drugs so much more efficient. And you can see the results. We're talking about faster discovery of new drugs for everything from cancer to viruses. It means we can design better versions of the drugs we already have. It helps us understand the risks from pollution, and it's even letting us explore totally new drug targets that we didn't even know existed before. So if you take one thing away from all this, let it be this. These computational tools have completely changed the game. Drug discovery is no longer just about trial and error. It's becoming an era of intelligent, predictive, and targeted design. We've seen what's possible today, which is already amazing. But it leaves us with one last really exciting question. With things like AI and true quantum computing just around the corner, what are the next generation virtual labs going to be able to do? What cures are they going to unlock?